This topic is based on DNA data storage. Many of our resources comes from the Missile Lab at UW. Their main focus is solving problems using ideas from both computer science and synthetic biology. We will receive support from the Missile Lab through Chris Takahashi. As the amount of data we produce grows exponentially, more efficient ways of storing and accessing information are necessary. One possible data storage avenue is to utilize nucleotides to encode information, similar to modern computers encoding with bits. Why should we use DNA data storage? Using current storage technology, the Earth will be covered in data storage facilities in the next 50 years. Our everyday silicon-based computers will normally last around 10 years before the data stored on it becomes corrupted and unreadable. Comparatively, DNA can last for hundreds of thousands of years before degrading, which makes it the perfect medium for data storage. DNA is also more efficient and faster at copying multiple files at once and is extremely space efficient. All of the data stored in a single data center can be stored in this spoon.
So what are the steps for DNA data storage? The first step to DNA data storage is encoding, which means converting data files from binary to the 4 nucleotides, the A's, T's, G's, and C's. These nucleotides are stored in the form of DNA, so hundreds of these DNA strands that encode for DNA files can exist in a single test tube. When we need to access the data stored in the DNA, we can use random access. Random access allows us to retrieve a single DNA file from the sample, and this will be explained more in the next slide. The extracted strands will then be placed in a sequencing machine and decoded back into bytes. PCR helps us with the random access process because it increases the number of desired DNA strands out of the thousands of strands in the test tube. By increasing the fraction of the desired DNA in the solution, the probability of us extracting out our desired strand increases. Therefore, PCR allows us to easily isolate our chosen strands for sequencing and decoding.
So for our project, we want to optimize the process of DNA storage. Some potential pieces of this are extractability, so the ability to access files from the DNA pool. One way we do this is making sure we use a PCR to produce copies of DNA to have access to files. At the same time, we want to optimize this process. We don't want to make too many copies of files because that will take up resources, but we want enough to where we can still access files. And finally, accuracy. We want to make sure that our data is not corrupted and that we don't lose any access to files. So our solution to this is to build a piece of software that can determine the optimal number of file copies to maximize the efficiency of DNA data storage and access. And outside of just that piece of software, we also want to create a synbio design that will block mutations when data is stored in cells.
Now, we'll talk about what each sub-team will be doing throughout the course of the project. So for Dry Lab, the largest task is going to be solving the optimization for the modular simulation in how many copies of DNA would be optimal to produce through PCR during file extraction. To do this, it's going to be gauging the probability of extracting the correct file versus the resources that will be expended through making copies of DNA through PCR. This will involve likely creating an index to determine the quality of an extraction event, which is going to involve also determining what sorts of resources and valuing those expenditures versus the optimal probability of extracting the correct file. The second thing that Dry Lab will mainly be focusing on is the process of magnetic bead extraction, both simulating this process computationally so that we can demonstrate the effectiveness of the method, as well as comparing the performance of magnetic bead extraction versus our optimized version of PCR to extract the file in the modular simulations. Next, going to wet lab. Although the optimization is primarily computational, the in vivo aspect of the project is going to be primarily wet lab focused. And for this, the focus is going to be, since storing DNA in vivo, or data in DNA in vivo, is going to mutate after lots of successive generations of the thing that we store the data in. The main goal of wet lab is going to try and prevent mutations through a variety of methods. And the second thing that wet lab would be focused on is performing experiments to verify that the optimization in dry lab is correct. There is the data that would be used would be likely provided by the missile lab, but there may also be synthesizing DNA involved. And going into human practices, for HP, there's a really large variety of potential ways to look at the HP aspects, and there's likely going to be a two-pronged approach of looking at the implications of DNA data storage technology, which has the potential to be huge, with a wide amount of applications towards security and encryption, data storage being more efficient, and preventing that problem of the entire world being covered in data storage facilities. So there are lots of stakeholders with that aspect, as well as integrated human practices opportunities when it comes to R-specific optimizations and preventing mutations with in vivo data storage. And for this aspect of the optimization itself, there would be more stakeholders with research labs focusing on DNA data storage and synthetic biology, because the resources expended through the PCR process is quite expensive, so optimizing that would save lots of money. And
so now let's take a look at what the project timeline for this project topic would look like, particularly when we have 3 different project aspects. Each of these aspects, as described in this timeline, roughly depends on the access to lab space. The nature of this project allows us to first focus on the computationally oriented aspects that don't require wet lab data. Now, modular simulations will be a very dry lab focused aspect of the project, as we'll be creating a simulation to optimize the number of copies of DNA to produce in PCR. A wet lab verification is necessary, but we'll do so when the labs open again. So for most of this summer, we'll be dedicating our time to these modular simulations. Following this will be the magnetic bead extractions aspect of the project. Here we'll be modeling and comparing magnetic bead extractions and the method to PCR. This is extremely exciting because it's a novel method to extract DNA samples from a single strand, and its potential is perhaps being 20 times more efficient than the current PCR method. This section will have an even distribution between dry lab and wet lab work. And just as soon as labs begin to open again, wet lab can begin working on this topic as well, while dry lab can begin this aspect beforehand. So we expect this to be following through autumn quarter and moving into winter quarter. Lastly, the preventing mutations in vivo storage aspect of the topic will provide wet lab an opportunity, a really unique opportunity to build a synthetic biology aspect to this otherwise very computationally oriented project. It'll even allow for a very project-focused biobrick design. This biobrick will prevent the occurrence of mutations in DNA within living cells. And this is, as mentioned earlier, intended to avoid corruption of data that are stored in the DNA. We want to emphasize that this idea hasn't been thoroughly researched yet in regards to its feasibility of success, but wet lab will have a lot of flexibility and have a creative process in deciding how to incorporate a biobrick in this project. And it's important to note that this aspect of the project topic doesn't necessarily have to be the last thing to get started on, as wet lab can begin thinking about the biobrick design starting this summer when the labs are closed. And once labs do open and the first 2 aspects are slowly completed, the project can slowly shift towards this last category.
Now let me just give a rundown of the iGEM deliverables in relation to the gold medal or special prizes. Since this project is very computationally focused, we have a nice shot at the computational side, computational special award. And because we're modeling a lot of different aspects, we have that criteria for a gold medal. And also, as Jason mentioned, we have very unique opportunities for human practices, which could lead to a unique take on integrated human practices. In regards to our possible iGEM tracks, we will most likely be pursuing information processing or foundational advancement. A little bit more work needs to be put into deciding which one, however.
So in conclusion, we strongly recommend continuing this project, and we believe it's well suited as an iGEM product, especially as a 2-year project. Some pros associated with this project is it has a very flexible timeline. We have identified an enthusiastic PI and have support from the Missile Lab. There is a large amount of versatility in many team aspects, specifically human practices. And it won't be heavily impacted by lack of access to the wet lab. Some cons associated with this project is we will have limited DNA samples from the Missile Lab, and it can be expensive to make our own. And then the computational work could take as little time as 6 months, but it could also take a very long time. So we want to complete our goals early.
Thank you.